The kingdom fungi is divided into various classes, which includes Phycomycetes, Deuteromycetes, Ascomycetes, and Basidiomycetes. In Phycomycetes, the mycelium is aseptate and cenocytic. In Deuteromycetes, Ascomycetes, and Basidiomycetes, mycelium is branched and septate. Now, let's talk about type of septa. In Phycomycetes, septa is absent. In Deuteromycetes and Ascomycetes, simple septa is present. Whereas, in case of Basidiomycetes, dolichopore septa is present. Some members of Phycomycetes can have cellulosic cell wall, whereas all members of Deuteromycetes, Ascomycetes, and Basidiomycetes have chitinous cell wall. In Phycomycetes, asexual reproduction takes place by motile zoospores or by non-motile aplanospores like sporangiospores. In Deuteromycetes and Ascomycetes, the asexual spores are conidia produced exogenously on the special mycelium called conidiophore. Conidia on germination produce mycelium. In Basidiomycetes, asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sexual cycle in Phycomycetes, Ascomycetes, and Basidiomycetes involves plasmogamy, which is fusion of protoplasms between two non-motile or motile gametes. Plasmogamy is followed by fusion of two nuclei called karyogamy, and karyogamy is followed by meiosis in zygote resulting in haploid spores. In Phycomycetes, the fusion of two haploid cells immediately results in diploid cell. However, in Ascomycetes and Basidiomycetes, an intervening dikaryotic stage occurs. Such condition is called dikaryon, and the phase is called dikaryiophase of the fungus. Later, the parental nuclei fuse and the cells become diploid. An important point to remember is that in Basidiomycetes, sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. Deuteromycetes is commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. The site for karyogamy and meiosis in case of Phycomycetes is the diploid oospore or zygospore. In Deuteromycetes, as sexual reproduction is absent, therefore there is no karyogamy or meiosis occurring in them. The site for karyogamy and meiosis in Ascomycetes is ascus, and in Basidiomycetes it is basidium. The sexual spores in Phycomycetes is zygospore. In Deuteromycetes, sexual spores are absent. In Ascomycetes, the sexual spore is ascospore, which is produced endogenously in the ascus. In the Basidiomycetes, the sexual spore is basidiospore, which is produced exogenously on the basidium. Fruiting body is absent in Phycomycetes and Deuteromycetes. In Ascomycetes, the fruiting body is called ascocarp, and the fruiting body of Basidiomycetes is called basidiocarp. Examples of phycomycetes include Phytophthora infestans, which cause late blight of potato; Albugo candida, which causes white rust of crucifers; Rhizopus stolonifera, often called the bread mold; and Mucor, which is commonly known as pin mold. Let's discuss the examples of Deuteromycetes, which includes Alternaria solani, that causes early blight of potato. Other examples include Colletotrichum and Trichoderma. Examples of Ascomycetes is Neurospora, which is used extensively in biochemical and genetic work. Other examples of Ascomycetes are morels and truffles, which form edible ascocarp, and claviceps, which causes ergot of rye. Lastly, examples of Basidiomycetes include Ustilago, that causes smut; Puccinia, which is infamous for causing rust; and Agaricus, which is the common mushroom.